This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 156, recorded November 4th, 2011. Hey everybody, I'm Vincent Racaniello, and this is TWiV, the podcast all about viruses. Joining me today from North Central Florida is Rich Condit.
Hi, Vincent.
How are you, Rich?
I'm good. How are you?
I'm pretty well. Have you had a good week?
Oh, yeah. It's a great week, you know, especially when it rolls around to Friday. Actually, today, get this, today at the University of Florida is a holiday.
What's the occasion?
Homecoming.
That's funny.
This is serious.
So no classes?
No, nothing. It used to be just no classes. Now everybody, you know, because since there weren't any classes, everybody took the day off anyway, so they decided to call a spade, and it's a holiday.
Nice.
Except here I am.
There you are, and your lab is still cranking away.
Well,
probably.
I'm having a good time.
Hey, I had a good time with your buddy Grant in Brazil last week.
Oh, good.
We had a nice time.
Good.
It was great.
And Nassim had a good time.
Nassim too.
Yeah, Nassim and Clarissa and all the gang.
Clarissa, actually the Brazilians were great. I had a great time. I felt really loved. It was really good. And they were friendly. They're big TWiV fans. And I had a great time. I wish I could have stayed longer.
I listened to the TWiV. It was a nice panel.
It was good. We had a bunch of good people, yeah.
Cool.
Alan Dove cannot be with us today because in the Northeast, last Saturday there was actually a storm. We had snow here, and the trees are full of leaves, and the branches broke off, and Alan has had no power all week.
Does that mean that Alan himself doesn't function? I mean, can you put batteries in him or something?
Alan's always got power.
Oh, okay.
But we do have someone in the TWiV studio today to join us. None other than Dickson Despommier.
Hello, Vince.
Have I kept you in suspense?
That was great. That was really good.
I tried not to laugh at all these horrible jokes also. So
I didn't tell any jokes.
Well, you were waxing humorous.
How have you been, Dickson?
I've been very well, thanks, Vince.
You have been in China.
I have.
How was it?
It was great. I just got back from Shanghai. And,
were you Shanghaied?
I was not. Well, actually, I was Shanghaied. I was enamored with Shanghai. We were there exploring the possibility of vertical farming in China.
Cool.
They're very hot on this topic right now.
Great.
Yeah.
All right. Well, it's good to have you here, Dickson.
Wait a minute. While I was there, I picked up a rhinovirus. Maybe you guys can tell me what kind.
Well, how do you know it was a rhinovirus?
Well, it's making my nose run on all kinds of stuff.
There are other viruses.
Well, I'm sure. All right.
Don't assume, please. If you
I
would
sure.
like, we could swab your nose and culture it.
You know, I thought about it.
You go ahead and do that, Vincent.
I don't want to. I don't want to get near your nose.
I don't want you to get near my nose. How's that?
I'll give you a swab and you could stick it in your nose
That'll
yourself.
work.
How's that?
That'll work.
I'll swab
I can't
it.
believe that Alan has been out of power for a whole week, man. That's
more.
really something.
Yeah, that's right. And Connecticut, too, by the way. Western Connecticut. Awful. These trees fell right across the power lines, and they don't have as big a power companies up there as we do down here, so they have a more difficult time repairing everything.
That's what you guys get for living in a miserable climate.
It's not supposed to snow when there are leaves on the ground. I tell you, I walked outside, and you hear gunshots. The branches were snapping left and right continuously. We're lucky they didn't break our wires.
Right. By the way, Rich, tell me that next time a hurricane hits down there.
Yeah, right.
Or tornadoes.
Before we get on to our stories today, I want to make a little request of the TWiV listeners. Now, going forward, we would like to do things with TWiV that require some funds, and we plan to raise them by advertising. And we have some advertisers who are interested, but they would like to know who our audience is.
Aha.
So our friend Mark, who used to work for Drobo, he was one who got us the Drobo contract originally. He has a new company, and he actually has set up for us a listener survey.
Oh, my.
To find out who you are, because advertisers want to know who you are.
Right. Whether you'll buy their products.
Yes, because we have a bunch who ask us questions, and we can't give them much information other than you love TWiV, and that's not good enough for them.
Right.
So we have a website that you can go to, triplemojo.com/TWiV. It's all one word, triplemojo.com/TWiV. It's a survey. It's a couple of pages. It'll take you 8 minutes. Please do it. And that will really help us get our demographics. We're going to run this for about 2 months, I think, so we can get everyone. All right, please do it. I don't want any money from any of you, but we do want our advertisers to support us. So
Yeah, there's a
please.
lot of listeners out there, according to the surveys. So if everybody could really help us out by doing this.
Yeah, we want to keep TWiV and TWIM free always.
And if you act now, we'll send you 2 plus shipping and handling.
And we think that the model of supporting it by advertising will work. So please take the survey, and that'll send us on our way to getting advertising support. All right. 3 stories today. Maybe we'll get to all 3. I don't know. First one is a paper that just came out in The Lancet. Efficacy and effectiveness of influenza vaccines, a systematic review and meta-analysis. From Osterholm, Kelly, Summer, and Belongia. And a meta-analysis. Dickson, do you know what a meta-analysis is?
I do.
What is it?
It's when you take metas and you analyze them.
Good, Dickson. Thanks.
It's so useful.
It's
I know.
it's past existing data that you've just remodeled and
Yeah.
re-examined.
There you
Of course, you don't have to collect the data; it's already been collected. Of course, I know what a meta-analysis is.
All right.
Come on.
Actually, I didn't know the formal definition of a meta-analysis. I knew what these guys were doing, but I didn't know that.
Yeah.
Now I know.
Oh, okay.
Isn't that great?
It's a database that exists. You just put it through another way of looking at it.
Okay.
So here they wanted to know how good is the flu vaccine.
Right.
There are actually 2 of them. There's an inactivated one and an infectious one, right? You put it in
Right.
your nose. And they start by searching the literature. They found since 1960-something, 5,707 articles about various trials of flu vaccines.
That's a lot of meta.
So there are 2 kinds. They're placebo-controlled, double-blinded, where you give a group the vaccine and you see what happens. And then there are observational studies where you take information. Were you vaccinated? Did you get sick, et cetera?
Right. The problem here, Vince, is, if you don't mind me just starting to interrupt,
I don't mind at all.
is that there have been so many different versions of the flu vaccine. How could you possibly be able to compare them all?
Well, that's why there are 5,707
No, that's right.
articles.
So how would you know which one?
Well, they pulled out the ones they wanted.
Yeah, they have a very stringent screen here for what studies are acceptable to them. Okay, now
I'm not questioning the studies, Rich. I'm just questioning the fact that you've got a moving target with regards to the vaccine itself.
Well, there are
Right.
2 different kinds of flu
It's
vaccine.
like
Dickson, there are
whooping
not that
cough.
many. There are a lot of different kinds of people. That's probably more of a problem. But
And
they're-
different manufacturers.
Well, that's true, but they're supposed to make it the same way.
And
Well, there's only
Vince-
one manufacturer of the infectious vaccine. Okay?
Only one.
Yeah, MedImmune. And then the other one, the inactivated, is made by a lot of different people. So your point is well taken that there could be differences.
and they did not take that into account.
They did not,
Nevertheless.
No, you just made some assumptions.
But they narrowed that down to 31 for this paper. They only looked at 31 studies out of 5,707.
It's amazing the chart of the sort of screening procedure that they used.
Yeah.
So the main criteria, there were a number of them, but the main was to confirm infection by either viral culture or PCR.
Right. And that's a very stringent criteria.
Absolutely.
All right, especially since PCR only existed since the 80s.
Exactly. So 1967 studies are
Yeah,
out.
all of the studies they list them. All the studies are pretty recent. Let's see, from 2006 to 2010, or there's a couple here from 2003.
Yeah.
But they're all pretty recent.
The interesting thing here is that they threw out all the ones that use serology to confirm infection. That is, take serum and look for antibodies to the viral hemagglutinin.
Because if you've been vaccinated, you should have antibodies already.
Exactly.
Dickson, you are just right on.
Right on the money.
Yeah, I haven't been here in a while. I haven't lost anything here. Come on, kids.
No, you seem to have gained some weight.
That's unkind and unfair and probably true.
Dickson, I love you. I'm not unkind. What were we? That was a great point.
We're talking
I loved
about
it.
flu.
That was great.
Yeah, we were.
You're absolutely right. So the serum antibodies are not
way to
easy
do that,
because if you immunize, you're absolutely right. You get. Okay, so they threw out all those, focused on the 31, and the conclusions are very simple. All right, the inactivated vaccine, 59% efficacy in adults 18 to 65 years old. That's the only age group where they had data. Like 2 to 17 years, no data that they looked at. And 65 years or older, no data. Only 18 to 65
Question.
years. And you're not in that range,
No,
Dickson.
I have to tell you this. No, I'm out of that range. You're absolutely
And
right.
soon I will be too, and
But
soon
there's
will Rich.
A lot of people die from what they would call flu-like or influenza-like syndrome. Older people.
That's right.
How about the autopsies, or if there were autopsies, or taking samples to see what they died from actually?
Well, that's another kind of study. I mean, in this
Right,
particular case, they were
Yeah.
looking at people that they knew got the flu vaccine and
Sure.
doing PCR and that kind of stuff.
Yeah. But does that data exist also? I mean, is that available?
I don't know. You want to do the meta-analysis?
The
Well,
thing is, Dickson, it's
CDC probably has that.
from my understanding, on the death certificate, it's very rare to put flu as the cause of death. So it's hard to tease out that information.
They call it pneumonia.
You would have to.
Yeah.
Do PCR.
Right.
I don't know if the data exists, but that's a different story because here we're looking at live people.
Sure.
So 59% efficacy. What does that mean to you, Dickson?
That means it's not good enough.
It's pretty lousy, isn't it?
It is lousy.
But that's an average because there were some studies that were better and some worse.
Do you think that represents genetic differences in human populations?
It could partially. I think it
And
also
age
is
group
because the
differences?
vaccine isn't very good.
What if you broke it down into finer quantities of age groups?
In fact, that's the problem, that the vaccine is broken down. Try the veal.
He's here on Wednesdays.
Because it's broken, it doesn't induce
Right.
a really good
Right.
immune response.
But do you think there's any difference between 17-year-olds and 65-year-olds?
Yeah, I do.
I do too.
Yeah.
So I would like to see that data more finely expressed.
Yeah.
Right.
Broken down. Yeah, I don't know if they can do it.
You know what the definition of a biostatistician is, don't you?
What's that?
It's a scientist broken down by age and sex.
Okay. That's very good, Dickson. All right, 59%. That means, Dickson, if 100 people get the vaccine,
Yeah,
only 59 of them would be theoretically protected, right?
exactly.
That's for the inactivated
That's
vaccine.
inactivated.
Yeah, that question here also means, what does it mean to be protected? Do you get sick but you don't get ill?
It's a good question. Well, they
Are you
didn't
getting infected but not sick, is what I'm saying.
Well, in this study, the ones that they teased out for this meta-analysis, it would be there is no virologically confirmed influenza.
Right.
Okay, because that's the studies they pulled out had to fit
Right,
that criteria.
Yeah, they basically, and this is what you raise here to me is an important aspect of this is that there's no attempt to address whether they got sick or how bad their symptoms were or anything like that, because it could be, and I mean I don't have any data, but it could be that people who are vaccinated and wind up PCR positive nevertheless have a much milder disease.
Mm-hmm.
It could be that it actually impacts on mortality. We just don't know. The data
Yeah,
aren't
it's a
there.
good point.
Those are the same questions we rose with regards to a TWiP episode we had for the malaria vaccine that just was announced. The efficacy of that was about the same as this. And it didn't prevent infection, it prevented disease.
That's right.
So I would like to know whether this prevented infection or disease in this case also.
So all we know is, I'm not sure that they recorded that for these studies, but what we know is, PCR or viral culture. Now, the infectious vaccine, 83% in children 6 months to 7 years old. Boom, that's it.
Yep.
That's the
Right,
only
and
age
that's-
group.
That's the only one they analyzed.
That's the only one they
It
analyzed.
said
That's
that
the only
no
one
trials.
got-
How interesting.
Yeah, no trials met inclusion criteria for any other age.
How interesting.
There have been other trials for other age groups and stuff, but they don't meet the criteria of this meta-analysis, which is the PCR positivity. There's other criteria as well.
So are they suggesting that these studies need to be done in order to verify the efficacy of the live viral vaccine?
Yeah, I think, yes. In some places, they suggest that some of these studies should be done. One of the problems is they raised the notion that there's ethical issues, in fact, in doing studies like this in people who are over 65.
Yep.
Okay.
Yeah, it's not ethical. Can't do a placebo-controlled trial in 65 and older.
Yeah, because they're more susceptible to the fatal effects of the virus. I understand that part, but they could still fill in the missing gaps. I mean, that's a big age group missing.
Well, I think because the infectious vaccine, that's the one you put in your nose, flu mist, 83%, bless you, if that were shown to be that good in other age groups, you could switch. I mean, right now they say here that only 9% of the flu vaccine in the U.S. is this attenuated vaccine. So you could increase it, but you'd have to know that it's better in other age groups.
Well, it begs those studies then.
They do say something here that's interesting. They say, I don't know if I can find it, that if you have a licensed vaccine, you can't do a placebo trial. Where is that?
Because it's already been done?
Oh, you have a conflict of interest.
Here it says placebo-controlled efficacy studies. That's one where you give a placebo to some, are not feasible for licensed vaccines.
Because they've already had to go through that.
Not feasible.
I think that you could probably do it for a different age group because that's what they did for flu mist. So, say flu mist, if it hasn't been properly trialed with these criteria in over 65, you could do it. There's actually one study here that they report over 60 years old, but they got no efficacy. It's 42% efficacy. They say it's nothing, and it's not licensed for that age group in the U.S.
That's interesting. The very group that you're out to protect and to show efficacy for, they're not allowed to do the studies. That's really crazy.
So superficially, it looks like, at least superficially, it looks like that the live vaccine is more effective than the inactivated vaccine.
Correct.
However, they're in different age groups.
That's right.
Rich, you can't say that because they didn't do the efficacy studies for the killed vaccine in that age group.
Exactly.
So now you're stuck with these two disparate results.
Right.
That you can't put together because you don't have the missing data.
So you can do an observational study. You could say give people over 65, give them all the infectious intranasal vaccine and see how they fare, right?
What's the health risk from the live virus vaccine?
No, there's no risk.
Okay. They discussed this to some extent. In the U.S., that vaccine is not licensed for people over 50 years of age. However, in Europe it is, and I don't know of any problems with administering that vaccine in Europe to people who are over 50.
Was it waiting for FDA approval or something?
I suppose.
I think because the studies haven't been done.
Right. In order to get it approved here, you have to have the right studies done.
And Europeans don't count in this case.
No. And they're not going to do a placebo-controlled study.
It reminds me very much of this. What happened for introducing metronidazole into this country? It's a drug that's used to treat some parasitic infections. They'd been using it for years in Europe to treat trichomoniasis, which is it's called Flagyl. But the USDA would not approve this drug. And finally someone said, look, all the clinical trials in the world have already been done. All you have to do is go to Europe and see the results that they've got already. And they did, and they approved the drug.
That kind of thing, I suppose, could happen with this.
Right.
It hasn't with flu mist.
No.
So I
if
think
you could travel to Europe, would you take it, Vince?
Flu mist?
Yeah, I'd take flu
I
mist,
would.
sure.
Yeah.
My kids get it because they're in the approved age group.
Right.
But I'd take it. I think it's probably the better vaccine because it's replicating.
Yeah,
right.
So there's a couple of issues about this, and it's worth studying, worth discussing. One is that it's replicating. The other is that it's administered
via the route that
the route that the infection normally happens.
Exactly.
Okay, so you're going to induce theoretically mucosal immunity, which is going to be the more important barrier.
There you go.
As opposed to the circulating antibody type.
So this is like an IgA response that you're talking about.
Mm-hmm. In fact, I always had problems with making humoral antibodies to flu to protect the respiratory tract.
Exactly.
Right. I've never understood how that works, basically. I guess some IgG gets into the lungs.
Correct.
Through the circulation.
No, you're absolutely right. It does.
But it's not going to be, theoretically, it's not going to be the most effective way to do this.
No, of course not.
Maybe that's part of the
And I think that was a major motivation behind developing flu mist to start with.
Sure.
Yeah. So
maybe AIDS vaccines should be introduced via the urogenital tract.
Well,
I'm sure that's a consideration.
I'm sure. So what do we think of this report? What's it going to do to immunization?
Well,
it generates further studies.
What about, people who are on the fence already, I'm not sure I need a flu vaccine.
Yeah, right. They're going to say no.
Because the press has picked this up, as you know.
Right.
If I were that age group that I was, and I am that age group actually, do I get a flu shot every year? No.
Do you?
I don't get a flu shot every year.
Now and then?
I've gotten the flu shots in the past.
Do you get flu?
No. I don't think I've ever had the flu.
You don't want it.
I know.
Boy, it doesn't.
No, I hear you.
I
hear you.
Jeez.
I do hear you.
I mean, I had a respiratory illness once that I was not diagnosed as flu, but it takes a lot to knock me out, and I was knocked out.
No, I've seen it happen too.
Yeah.
In fact, I know a tragic event here. One of our faculty's sons actually died from flu at the age of 2 years old.
Wow.
So he gets a flu shot every year, of course.
So I think that it might have a negative impact on immunization coverage. It shouldn't, because in the ideal world it shouldn't, because there's no downside to getting.
Couldn't hurt.
That's right.
It can't hurt, and it can help. It's the best we've got.
That's true. You go to O'Hare Airport and it's there free. You can just put your arm out and they'll give you a shot.
Yeah, they say here in the article that it's the best we have. We should use it until we have something better.
Well, there you go. Well, there might be something better.
Unfortunately, that's not a good endorsement, right?
No.
But,
the nasal live vaccine is a better choice if you could have it.
Yeah, you can't. Not all of us can have it. Trina Suderos wrote me an email this morning, and she said, isn't it better if at least you get your whole family immunized and at least you don't transmit it amongst yourselves?
If the vaccine works, yes.
Yeah, but 59%, right?
So half of your family members will get it and the other half won't.
Yeah, she said basically your family and at work, everyone should get it. The more people, the less likely it is
herd
to be
immunity is what
transmitted.
she's driving at, yeah. Sure.
But the problem is, so she wanted to know if there are any numbers about herd immunity, and I'm not aware of any.
Well, there's theoretical numbers, of course, but
the problem is you go on the subway and someone's sneezing in the car, and your efforts to immunize yourself and your people at work fall into the subway tracks.
We just got back from Shanghai. My wife and I took the subway every day to various parts of Shanghai, and I'm sure this is how we acquired this whatever this is, infection that we have, a respiratory infection.
Yeah, in the public. Also, you changed locales. That
of course,
contributes as well.
Of course.
Interesting
study.
It was
inevitable.
Usually the coverage that is required to induce good herd immunity is upwards of 80 or 90%.
Yeah. So this wouldn't.
So I think this falls significantly short of that.
That's
Depends on the virus. Yeah,
that's right.
It depends on the virus.
I was thinking about West Nile virus and how it swept through this country, and now we've got reduced rates in endemic centers like California and Texas. So you might have herd immunity working in those cases from the exposure to that virus infection.
At any rate, this is an important paper, because it's a well done study. The criteria are very stringent, and so that has to be taken into account. But it's important to have these kind of numbers out there. They provide motivation to improve the vaccines that exist, or
I think that's the key.
Vaccines.
Yeah, we need better vaccines, and they're certainly being worked on.
What about the vaccine that uses a core protein that's common to all the influenza strains?
Yeah, it's being worked on.
Yeah, has any data come out from that yet?
There are some, but not trials yet. It's still experimental.
Yeah, it's in very early stages.
One of the main criticisms of meta-analyses is that you tend to lump crappy data in with the good data.
Yeah.
But here they've tried to tease out of 5,700 down to 31. So I think it's a good study, and I think the number 59%. That's it. If someone wants to know what's what's the effectiveness, there it is.
Yep.
You have it. I think people have to make their own decisions, but I favor getting immunized to reduce transmission.
Right.
Because it does kill people.
Yes.
And what's the point if we can prevent it?
Exactly.
And both you and Alan have written blogs on this. I think we ought to put both those links in the show notes.
And also our friend up at Vassar has written a post, right?
Yes. It was, David Esteban. It has a nice little blog on this. It was actually before the paper came out. He was responding to, he said there's a photo circulating on Facebook that shows the package insert for a flu vaccine that appears to indicate the vaccine has not been shown to be effective against influenza. And, of course, this creates a lot of stir.
An insert in a vaccine package that says you don't work?
Because they talk about what actual trials have been done.
Oh,
and the thing is, you can get a vaccine approved just by showing that you induce a certain level of antibody response.
You do no harm, at least.
Okay?
Yeah.
Well, you get a certain level of antibody response, and they'll say, okay, the vaccine works. But that doesn't mean that you've done a clinical trial to show that it's effective against flu. And so David sort of takes this apart and goes through all of the different criteria that can be used, and actually lists all of the different makers of influenza vaccine now, and has a little comment on the extent to which those vaccines have been studied. So there you go.
All right, we'll put links to all three.
Good.
All right, the next paper is a Brevia in Science, one of Alan's favorites, one-page article.
Those are easy to absorb and remarkably complete.
Compatibility with killer explains the rise of RNAi deficient fungi.
Ooh.
Dickson, is it fungi or fungi?
I think it's fungi.
Thank you, sir. This is a
Remember,
really
I'm
cool.
a parasitologist, not a mycologist.
But you know the words.
I do.
Drinenberg, Fink, and Bartel. So this has to do with yeasts, Saccharomyces candida, and a variety of others. And many of these have...
This has to do with beer. This is important.
Beer.
Yeah, very important. Don't forget the wine, too. I mean, I think wine falls
Right.
So it turns out that many of these fungi don't have RNA interference. Did you know that, Dickson?
No, up until this point, I wasn't thinking
I wouldn't
much
expect
about
you
it.
to know it.
Probably not on the top of your hit list.
Not on the top of my hit list.
You do know what RNA interference is,
I do,
right?
of course I do.
It's this wonderful system where RNAs are chopped up and then used as templates to chop up more RNA. So it's like an innate immune response.
Sure.
So
Almost
it turns
like
out that baker's yeast doesn't have RNAi. Saccharomyces cerevisiae.
Amazing.
But you can restore it to Saccharomyces by putting in Dicer and Argonaut. Argonaut. Jason and the Argonauts?
Yes.
Remember that?
I do.
And those are two
The
components
golden
of
fleece.
of the RNAi pathway.
In this case, it's golden brew. Uh-oh.
My phone was ringing. I had to pull out the plug. So they took the RNAi, the Dicer and Argonaut, from a related yeast, Saccharomyces castelli. They expressed it in Cerevisiae, and lo and behold, RNAi is back in Saccharomyces cerevisiae.
But you have another tag to this story, don't you?
Yeah, there is another tag. What's the outcome?
No. Isn't there a double-stranded RNA virus
There you go.
lives inside the...
Hey, Rich, had you ever heard of this virus?
No, I hadn't.
Saccharomyces?
I had. I had heard of it. Because, as a matter of fact, one of the guys who does a lot of the work on this killer virus, a guy named Jeremy Bruin, who was a colleague of mine in a different department at Buffalo.
Right.
And we did a paper of his not too long ago where he was one of the guys who first showed evidence of remnants of, what was it, a Bunya virus?
That's right, integrated
genome in the
in the
At any rate,
genome,
he's made most of his living working on killer virus.
So they wanted to know, why do some yeasts have RNAi and others don't.
Why is it called killer virus first?
Well, we didn't get to the virus yet.
Oh, okay.
Hang on.
All right.
Oh, yes, we did. We've been talking about it.
No, yeah, but we didn't really. We need to describe this virus.
Yeah, please.
I didn't know anything about this thing.
All right,
It's
so
really
we're
interesting.
going to do that. So let me just say one thing. So they looked at 50 different growth conditions, and they found that the one thing that was different when you put RNAi in is that you lose this killer virus.
Okay.
Right.
Now, why should?
Now let's talk about the virus.
Sure.
So
Why
these
should
two things are
Saccharomyces
mutually
cerevisiae
exclusive? You can either have RNAi or a killer virus, but you can't
have both?
That makes sense because the RNAi pathway will get rid of the killer virus because it's a double-stranded RNA.
So does the killer virus kill off all the other invading
Okay, so here's
fungi?
the deal.
There
This
you go.
killer virus
Keeps
is
the
amazing.
culture pure.
Yeah,
Double-stranded RNA genome,
Yeah.
about average 5 or 6 kilobases in length, encodes 2 proteins. So double-stranded RNA encodes 2 proteins that look for all the world like the arrangement in a retrovirus genome, with GAG and PAL, and a ribosomal frame shift to give you the PAL open reading frame. So this is I can't believe that this is any kind of convergent evolution. There must be some common ancestor between retroviruses and these guys that give this arrangement, because it's just too weird otherwise. At any rate, so this virus does not have an extracellular phase. It stays inside the cells, but it's encapsulated.
It's weird,
all right?
isn't it? Yeah.
And that makes, evolutionarily, that makes sense, because if you're going to have double-stranded RNA in the cytoplasm, you better hide it.
The capsid.
Otherwise, it'll trigger an innate immune response. So you hide it in a capsid. This is the same way that the other double-stranded RNA viruses work, like rotaviruses and reoviruses. They actually have a double shell. Okay, they're naked capsid viruses, double-stranded RNA in a double shell, and they infect basically using the outer shell, and inside the cell, the RNA stays encapsulated in the inner shell, and it encapsulates the polymerase as well as the RNA. So you make messenger RNA and extrude it from the capsid. So the only thing the cell ever sees is messenger RNA, and it's duped. Okay, and then what happens is you make capsid protein off that messenger RNA and polymerase and stuff, and you use those proteins to encapsulate the viral messenger RNA, and then finish it off and make the second strand inside the capsid. So once again, the cell never sees the double-stranded RNA. And in yeast, as I understand it, these things stay in the cell. They don't have an extracellular phase, and they're transmitted as the yeast divide, just as a particle that stays in the cytoplasm. So it's inherited.
So why are they called killer virus?
Ah, good question. So
If they can't do it outside the virus that
there's a satellite. There's a satellite, call it virus. I don't know if you call it a satellite virus. Is that a proper word?
They call it satellite double-stranded RNA. I think it's just
Okay. So it's
RNA.
a double-stranded RNA that makes a toxin. All right? And that thing is a double-stranded RNA. As I understand it, that's encapsulated in the same capsid. It uses the capsid
Really?
from the
There are 2
major
genomes
virus.
inside the same capsid?
No, you got
separate.
2 capsid separate genomes.
Okay.
Okay, so
But
the
the one-
killer RNA, it's kind of like Delta virus for
satellite. Yeah.
hepatitis.
So
Hepatitis delta. It's using the capsid from the other guy, and this toxin
Right.
can be secreted from the yeast and kill other yeast that don't have the toxin. The toxin also, by mechanisms not understood, there's a precursor to the toxin, makes the cells that make it resistant.
So if
Okay.
if you cure Saccharomyces cerevisiae of the killer virus and keep the satellite virus,
Can't.
Can't, because
because
it needs the capsid.
Well, these are
From the-
also the polymerase.
Also
okay, so the
polymerase, right.
the satellite virus doesn't encode anything except the toxin.
That's right.
I
Correct.
got you. I got it.
So if you take it out,
Okay.
and so presumably
Okay.
in nature these are
This
used
is
to,
weird.
to kill.
You bet.
com-
This blows my
competitors, huh?
mind.
This is weird because that means that brewer's yeast forever will be brewer's yeast, right? No, you're laughing, Rich, but I was at the place where brewer's yeast first began. You're laughing at me, I bet you are.
No,
but I'll
I'm
tell
not.
you,
I'm just,
it's called the Road to Jerusalem, and it's a pub, and it's in Nottingham, England, and it's dating back to the 11th century. And that was the first brewery in the New World.
cool.
And the yeast is all over the place. You can isolate it. It's still there. It smells like yeast. You walk down into a cave where this tavern is, and it smells like a yeast factory.
So I don't know if the brewer's yeast has the killer virus or not, but
No.
It does not?
No. Well, it's got it, but it doesn't have RNAi. Sorry. Yeah. So Saccharomyces does not have RNAi, and it and it has the killer.
It does. So is this how they keep their cultures pure? By keeping out other invading virus?
Well, some of the fungi have killer also, so they would be resistant. In fact, that's one of the things they did next is they sequenced whole genomes of many different fungal species.
Sure.
A ton of them here in Figure 1. How many there are here?
Goodness.
A lot. At least 20. And they could tell which have RNAi and not, and which have killer RNA and not.
So this has something to do with ecological biome territoriality or something?
The toxin?
Yeah, I mean...
I guess so,
in the wild, this sounds like
sure.
antibiotics and cholecines and all these other microbial
absolutely.
products that are...
Yeah, that's it.
Okay, this is fascinating stuff.
So they basically find that if there's RNAi, there's no killer, and if there's killer, there's no RNAi.
Right.
In these strains.
Right.
Now, they noticed, though, that all the strains that have lost RNAi have done so recently. And so they suspect that it's not a good thing to lose.
Evolutionarily recently.
Yes, exactly. They think that it's not good to lose RNAi.
Yeah, right.
Because they say there are no descendants because it looks pretty recent, right?
Right.
So they say, although compatibility with the killer system can explain the persistence of RNAi-deficient fungal lineages for many millions of years, lineages that lose this elegant transposon defense might be doomed to extinction over the longer term.
Goodness.
So I'm having a hard time here.
Yeah.
Why are these mutually incompatible?
Okay, so you mentioned that these are encapsulated double-stranded RNAs, but they have to make messenger RNAs.
Right, so the RNAi can get rid of the killer.
Exactly. So as soon as the messenger RNAs come out of the capsid, they're chopped up by RNAi, and eventually the virus goes away because it can't replicate.
But the killer isn't necessarily selecting against RNAi. It simply can exist where RNAi does not.
Yes.
Well, actually it can in a way because the killer provides a survival advantage for the yeast that carry it.
So a bigger question is, why isn't the yeast that carries the killer killed by the toxin that's produced by the satellite?
Yes.
And they don't know the answer.
They don't know. Yeah, that's a good question.
That's a great question. They don't know the answer.
The assay is very cool, by the way.
Yes, I agree.
You played out the sensitive cells on a petri dish, and then you then on top of them played out less numbers of the killer, and you get clearing.
Plaque assay.
Yeah, exactly. Wherever the toxin is, and then they show that basically when you put the RNAi pathway in, you get fewer plaques.
Maybe the toxicity of the toxin occurs after it's secreted. Maybe there's some conformational change that it undergoes before it's secreted that renders it inert.
There is a precursor form of these toxins. They're made as a precursor and processed.
So they're not toxic up until that point then?
Well, that could be, but there's more to it than that because the cells that carry the toxin are actually resistant to attack by the same toxin from elsewhere. Okay?
Got it.
All right, so it's more than just it not being active in the cell. They actually resist the activity.
And how does the toxin work in the cells that it does kill?
There's a bunch of these different toxins, and they work in a bunch of different
ways.
Of course. I was not able to research all that.
One of them is that makes ion. That makes channels in membranes, for example. Poke holes in the plasma.
Like Meganins, then.
One idea
Are they Meganins?
They're not Meganins. No, they're bigger than Meganins. They're bigger proteins. Meganins are small.
Okay.
One of the ideas is that the precursor to the toxin actually confers immunity, and then once it's cleaved, it's a toxin.
Okay,
There's a lot of...
Got it.
There's actually a very nice review article that you found, Rich, that deals with this. And there's a lot to learn here. It's quite an interesting system.
Great stuff.
New world for me, too.
And the evolutionary implications are very, very interesting
Amazing.
as well. So there you go.
That's fantastic.
And the summary of this is killer virus selected for fungi that lack the RNAi machinery. So that's basically the killer virus can only be established in fungi that lack RNAi.
Right. So if you knocked out RNAi production in a yeast cell that can make it,
We have to introduce the killer somehow to that yeast cell. Yeah.
then they would accept the killer.
They would accept it, yeah. You can introduce it
microinject
into
or
them,
something.
yeah. You can make
And all these have sexual stages, too.
yeah.
Right. I was just thinking the same thing. So you could acquire it, since it doesn't have an extracellular
Through
phase, or
gametes.
at least not obvious, you
That's
could
right.
acquire it by mating.
Yep.
These are really cool stories. We've had this for
Amazing.
a while now, but I think it's great because this
Yeah.
killer virus I've always been interested in, but I didn't know this RNAi story. Well, this is relatively new.
And I didn't. I've been interested in the killer virus, but I never really spent the time to find out how they worked, and it's fascinating.
So, if you, I mean, they give all these naturalist television shows. I'm so addicted to them. Like Life on Earth and Blue Planet and Planet Earth. And
The fuzzy animal shows.
they're great though because they show you all of the breadth and depth of mammals and birds and spiders but as you dig down through the layers of life to the microscopic layers, and you're in it right here, the complexity increases, it seems to me, rather than decreases. It looks like it becomes even more complex because the relationships are all based on chemistry and pheromones and microenvironments and secretion of inhibitors or attractors. It's a wonderful, fantastic world between the little grains of sand in a soil sample. And that's where all this is taking place. And, it's another universe. And if you look at the physics of our universe, and you listen to these crazy physicists, the way they're talking now is about the multiverses and all the origins of, black holes give rise to other universes now. These are micro universes, basically. If you were a microbe in that world, you would totally be unaware of what's outside at the layer of the soil and above, and even on that one planet. So it's it just allows you to dive into this stuff at every level.
And the other thing that impresses me is that, and I was thinking about this listening to the podcast that you guys did in Brazil, is that the more of this virus discovery that goes on, the more I realize that we are just swarming with viruses. I mean, there's probably just nucleic acid information being exchanged on a mammoth scale.
That's right.
Both in larger animals and in the microbial world that we've never really thought about before.
And Vince ate a pear for lunch today and told me, tell him what you told me, Vince, about your viruses in your small and large intestine.
Well, I was... this has to do with my pick of the week.
Sorry, I didn't mean to spoil it.
No, you don't need to apologize. I was eating the pear, and I was telling Dickson, I wonder how many viruses I'm eating.
Yeah.
Because I'm quite sure that all of our plants that we eat are full of viruses.
Yeah.
Sure.
And there's good evidence, because most of the virus in human feces is plant viruses, over 90% of the RNA viruses anyway.
Unless you're a strict carnivore.
That's right. I bet it would be different in a strip. Are there strict carnivores?
Oh, yeah.
Well, not among humans, not among animals. Right?
Okay, well, no, in humans.
There are people who don't eat any vegetables?
Of course. Name them, Vince. You know who they are. They're all Eskimos.
That's right. We talked about this once, yeah.
Yeah.
It would be interesting to look at their feces for the virome. Anyway, yes. We'll talk about this more in my pick. Should we do the third paper, Rich? I think we could do that.
You're the timekeeper. Yeah, we can probably
Yeah,
do
okay.
that.
I think you'll like this one because it's a pox virus.
Ah.
It's a paper published in Nature. Intravenous delivery of a multi-mechanistic cancer-targeted oncolytic pox virus in humans. So if you remember Grant McFadden, what do you need? You want to see the paper?
No, I need you to turn your screen from your laptop next to you until the left because the sun is shining in my eyes.
I'm so sorry, Dickson. Here I thought you wanted to see the paper.
Oh, I can see the paper
But you
from
didn't
here.
want
Vince.
sun in your eyes.
I can see it from here.
So Grant McFadden was on TWiV before his Brazilian time talking about his using myxomavirus to target tumors, and that's an example of using an animal virus in people. Mm-hmm. Here we take a human virus and modify it. And Grant actually said those are two of the ways that you make these oncolytic viruses. Mm-hmm. So, Rich, can you tell us about this virus?
Sure. First of all, I'm not sure it's fair to call it a human virus. It is a virus that will replicate in humans. But so this is, we've done this several times before.
Right. It's probably not of human origin.
You don't know what the reservoir is?
This is vaccine, right?
No, as a matter of fact. Okay, so that's a story in itself. So this is the this is the smallpox vaccine, right? This is Vaccinia virus. It was used as the smallpox vaccine.
Okay.
And if we've talked about this before, the story is that it was isolated from a milkmaid by Edward Jenner 200 years ago and used as a vaccine. There's probably been several different isolations since
cowpox or whatever.
Well, but that's the problem is that if you look, now that we can do this, and you look at the genome sequence, all the things that we call vaccinia that were used as smallpox vaccines, in fact, are not the same as what we call cowpox. Okay, though there was a paper published recently that was sequencing of a bunch of different cowpox genomes, and there's a lot more heterogeneity in the family of cowpox viruses that than we previously recognized.
How different is it from vaccinia?
Well, I mean, they're both orthopox viruses. They're serologically cross-reactive. If you do the phylogenetic trees, they wind up in distinctly different groups. Okay. But my guess is that there's a common ancestor in the not too distant past. But at any rate, so there's some questions about the origin of what we call vaccinia. Cowpox and other related viruses, the most common reservoir is rodents. Okay, and that goes for the virus we call monkeypox and et cetera. So my guess is that the natural reservoir for this thing was or is probably most likely rodents, but it has a very broad host range and can replicate in humans. It's and it can be transmitted from human to human. It only replicates by direct contact with a broken or abraded skin or in a mucous membrane. So you can transmit it from a human to a human by taking some of the virus and say from a lesion and transferring it to another one and to another person in a cut. And typically what it forms is this blistering lesion on the skin. So these guys start with, and this is Carolyn, would you pronounce that Breitbach or Breitbach? Breitbach, and a bunch of other authors, and senior author is David Kern.
Mainly from a company, right?
Yes, a company called Generex.
I wonder where that name came from.
Yeah, really. And
pandering to the
look
past.
Back in the history of this, I think the company was formed around this particular virus. This virus predates the company, as far as I can tell, by some period of time. The first descriptions of this virus were back in like 1999. So they started with the Wyeth strain of vaccinia. Wyeth was one of the manufacturers of the smallpox vaccine.
He just got bought by Pfizer.
Yes. And and actually, Wyeth is if you go to the Wyeth vaccine and plaque it out and pick a bunch of plaques, it's significantly heterogeneous. So this is probably a plaque isolate of some sort from that. I don't know the full history of this particular strain, but it's basically vaccinia. And what they have done is to clone into it, like I say, this was done back in the late 90s, clone into it, and importantly, they've cloned it into the thymidine kinase gene, two other genes under transcriptional control of the virus. One is GM-CSF, granulocyte macrophage colony stimulus factor, which is basically a cytokine and recruits all sorts of inflammatory cells and stuff to wherever it's being made. And the other gene that they've inserted is basically a marker gene, is beta-galactosidase, which will, for the uninitiated, it's an that will cleave a colorless dye into something that gives off color. So you can tell where beta-galactosidase is.
Yep.
So I think there's two important things about this. One, it was known at the time that they did this that if you inactivate thymidine kinase, the virus is attenuated in vertebrate hosts. That is, it in a model system like a mouse, you can arrange conditions so that if you infect a mouse with vaccinia, it'll die. But if you knock out thymidine kinase, the mouse will survive.
Right.
Okay.
Right.
So thymidine kinase, by the way, is an enzyme that takes a precursor to DNA, thymidine, that's not ready for DNA synthesis, and phosphorylates it, okay, which basically activates it so that it can now be used in DNA replication. So a virus that has thymidine kinase is better able to boost the nucleotide pools for doing replication. If it doesn't have thymidine kinase, it's less able to do that, and that's probably the mechanism for attenuation. So this virus, well, that's it. It's attenuated and it has a cytokine in it and a marker in it, and they've tested it as a candidate for oncotherapy. Now,
this is an interesting name. They call it a targeted and armed pox virus.
Like a drone.
So the
sounds like a drone going out over Afghanistan, and
the idea is quite similar to the rap that Grant gave us about myxoma, is that this thing apparently replicates much better, if not exclusively, in tumor cells as compared to normal tissue.
Mm-hmm.
Because?
Probably because the tumor cells have a number of signaling pathways constitutively active.
Yep.
They're on all the time. That normal cells don't have all the time. Plus, they're actively replicating DNA, so they're likely to have higher nucleotide pools.
Sure.
They say a lot of things in the paper about how this works, but the truth is, I think it's mostly empirical. All right.
I think they say that TK is often elevated in tumors, right?
Right.
Because they're actively replicating, yeah.
And there are other signaling pathways that are on in tumors that the virus needs to really replicate well in mammalian cells. And so a bunch of those things put together gives the virus a preference. It's not, I think it's the way they do this is to deliver it intravenously. So it has access to a number of different tissues. But even in an area, and the pictures are beautiful, in an area where there's tumor tissue surrounded by normal tissue, the virus replicates quite selectively in the tumor tissue. That, to me, is the most impressive part about this paper.
I'm confused a little bit, though, because in Virology 101, Vince and I discussed the first phase of infection, which is attachment. And there have to be specific receptors present, and ligands have to combine in the right way. So why does this virus target cancer cells rather than normal cells, unless it's got a receptor molecule which favors the attachment of this virus to those cells rather than?
My guess is it's getting into both types.
It is. So is this
doesn't replicate well?
Is that true for most pox viruses, that they will go into most cells, or
these orthopox viruses? I don't know about some of the others, but the orthopox viruses, most of the ones we talk about, part of the reason they have a very broad host range is that, they use very broadly distributed receptors. As a matter of fact, the receptors aren't really known. There's been a few attachment molecules that have been identified, and these are like sugars and proteoglycans and stuff like that are pretty ubiquitous on cell.
Because a lot of the pox viruses just stay in the skin, right?
Yes, they're actually that's not entirely true. Smallpox manifests ultimately on the skin, but initially it's transmitted as a respiratory infection, and goes through phases where it infects lymphoid organs, and then it goes through viremias, and ultimately erupts on the skin. So, in fact, it has a pretty broad tissue tropism, even in host.
Okay.
Vaccinia, the way it's ordinarily encountered, is transmitted by direct contact with the skin, and pretty much stays there. But I think that's mostly because for one reason or another, it doesn't really establish a viremia. Or that is, it doesn't travel through the blood well.
Got it.
So the purpose of this was to determine whether this virus, JX594, could infect tumors after being given intravenously.
Yeah, and I think the really important thing here is the selectivity for tumors given an intravenous route of infection, because a lot of the oncolytic viruses are targeted by actually doing intratumoral injections.
Right.
And that's not going to be practical for a lot of different tumors. But if you could administer one of these agents systemically, like through the bloodstream, and distribute it everywhere and get specific replication in the tumors, that would be a good thing.
Yeah, and particularly people with metastases, you can't possibly inject every one.
Exactly.
So you need to have something like this, right?
And I actually did a little calculation because I was a little concerned about the notion that I would imagine that this virus will infect both normal and tumor cells. That is, it'll get in and start replicating. But not go through a productive replication in the normal cells, but go through a productive replication in the tumor cells. So I would imagine that the normal cells that get infected, they're going to die.
Yes.
Right?
Yes.
So I actually did a multiplicity calculation, because at the highest dose they're giving these people about 10 to the 9th plaque-forming units per body. And what do we have? Something like about 10 to the 14th cells?
Yeah.
So it's a multiplicity of infection of 10 to the minus 5. So even if you infect some normal cells, it's probably not going to do any damage. But if you infect tumor cells and it replicates and spreads to the adjacent tumor cells, then you're in good. Then that's what you want to happen.
Right.
And it's going to do two things. It's going to infect this. It's going to kill the cells that it infects, and it's also, presumably because of the GM-CSF, going to recruit immune cells to the site and give a more robust immune response against the tumor.
But Rich, the virus produced by the tumor, are you worried that that's going to go on and kill some normal cells too?
Well, I wouldn't want... I'm not anxious to have this injected into me under any circumstances. Well, unless I'm, don't have any alternatives left. I would be worried a little bit about at least damage to the surrounding tissue. I would assume
that's right.
Would be a barrier to spread, because the surrounding tissue is not really going to support the replication well. But I'll tell you, as I said, the most impressive thing about this paper is the histological sections from the individuals that they treated, and they show really striking pictures.
Good targeting.
Tumor embedded in normal tissue, and they've stained it with antigen that detects the presence of the virus. And there's a it's robust replication apparently in the tumor, and nothing in the surrounding tissue, with a very sharp, very distinct boundary between the two. So that's impressive.
Yeah, they've achieved the goal of the study. They showed specific replication in the tumor.
Right.
Now, we should note that this is a phase 1, which means just see safety, and then
not efficacy.
Not efficacy, but they want to see if it replicates in the tumor. And these are people with advanced tumors that weren't treatable by any other means.
That's true.
So they said, look, do you want to try something? May or may not work. You may get another month. And they say, sure.
And that's typical for a trial. I think probably at phase 1 they probably present this and say, look, you're basically doing something. You're not. The chances that you're doing something for yourself here are real slim. You're doing something for us down the road.
That's right.
Okay.
So there were a variety of tumors: lung, colorectal, melanoma, thyroid, pancreas, ovarian, gastric, etc. So all different kinds, and in each one, pretty interesting that the virus goes right to the tumor, replicates extensively there. There are not bad side effects. They said they had some pustules, right? Which you might expect on the skin.
Right.
But otherwise, considering the doses. And then they said that some people, their metastases stabilized for the period, which, this is not an efficacy trial, but...
but to get that data anyway.
They got it anyway.
Sure.
But in some cases, though, there was stabilization without a lot of viral replication also, so they're not quite sure what's going on there.
How many people involved in each one of those?
There's 23 people total.
23 total. Okay, so not too many repeats.
It was an escalating dose trial.
Got it.
So they went all the way from
All right.
10 to the 5th PFU per kilo up to 1.5 times 10 to the 7th. So that's a more than 100-fold range in concentrations. And the greatest sort of effects that they saw in terms of detecting virus replication and stuff were determining was at the highest dose.
Yeah,
this defines the dose range.
They only got one infusion of virus. Okay, so that's probably artificial because you're going to want to treat multiple times if you're actually doing this. But that's the kind of trial this is. You get one dose.
So I think the take-home message from this is that first of all, it tells them where to look in terms of dosage, and it tells them that they are getting specific. It's not really targeting because, as you pointed out, Dickson, it's going to both normal, normal and tumor cells, but it's targeting in the sense that the replication is happening.
Got it. And that's because of the absence of thymidine kinase. Yeah.
That and the fact that the tumor cells have a bunch of signaling pathways active that the virus likes to use,
and most of those are kinases.
Mm-hmm. Yeah. And I think the other message is that it seems to be effective in a wide variety of solid tumors.
So, Rich, what do you think of this? They say all patients had a history of vaccination with vaccinia as children, and delivery was demonstrated in a patient with neutralizing antibodies present at baseline.
Mm-hmm. Interesting.
So this is always a concern with any kind of vector like this, is that it won't be useful in people who have an immune response, and you may not be able to reuse it. Well, first of all, if you're vaccinated as a child, and these people are all in their 60s to 80s, the youngest I think is 52. Although the smallpox vaccination is a lifelong immunity, it does wane a little bit, so these aren't as immune as you might expect. Right. But I also figure that probably the whole idea, they're probably overwhelming what immune response there is, because they're giving these people 10 to the 9th of these viruses IV. Boom.
It's a lot.
Okay.
It is a lot.
And so that really gives it a head start. Now, in terms of long-term replication in the tumor, I don't know. Apparently, it's not a problem. There's a lot of studies like this that I'm not familiar enough with to look. I mean, these pox viruses are used as vaccine vectors as well. And so this issue comes up again as to whether or not you can use it twice. There's a lot of studies that test this, but I'm not familiar enough with the bottom line on those studies.
I would think that if you give 10 to the 9th particles, you're going to get a nice, robust immune response.
Yes,
which they didn't measure here, but that's an issue they'll have to deal with.
That's going to yes, they're going to have to deal with
because you're going to have to give multiple doses.
And all I can say is that what little I know about the results of those types of experiments is that it's not straightforward. You don't get. You would kind of expect that if you do this once, you're going to get a big immune response that can be ineffective down the line, and I don't think it's that simple.
Well, a lot of these patients are being immunosuppressed anyway because of the cancer chemotherapy drugs they're getting.
Good point.
So maybe their immune system is compromised already.
That might help them, right?
Yeah, it could actually be helpful.
So this is very promising, and I think this whole area of viral oncotherapy is so impressive. I used to be very critical, but now we see this one. We see the work that Grant is doing. We had Replication talked about on this show, if you recall. There are a couple of other viruses in trials. So I think in 5 to 10 years we might be using. This one oncolytic virus is licensed for use in China already.
Really?
For certain kinds of tumors. So this may be useful.
I must say, however, after reading that book, The Emperor of All Maladies.
Did you finish it? You finished it.
What a terrific book. That is just absolutely one of the best books I've ever read.
Oh, wow.
And it gives me a really deep appreciation for how difficult a problem cancer is. It's really a difficult problem.
But it does end on a very low note.
Yeah.
I mean, he basically says cancer may be the price we pay for living.
Yeah, right.
Essentially. Maybe we can't do anything about it.
I think automobile accidents are the price we pay. If you go to any country where the the average age of the population is high, you'll find that most of those people are going to end up being killed in accidents.
Well, this fellow is Mukherjee has made the point that incidence is like 1 in 3 people, and it's creeping to 1 in 2 of cancer. 1 in 2.
Well, I think he throws in skin cancers there, which are curable. A lot of the cancers are
if you get
serious.
that high, if you get it quickly enough. Right?
Yeah, that's right.
Early
That's
enough.
right.
I just noticed that this same issue of Nature has a whole special section on autism.
Huh.
Oh yeah.
Yeah. I realize this isn't This Week in Autism, but might want to have a look at that. Okay, sorry.
All right.
Side there.
Email.
Okay.
Now, Rich, before we do email, you wanted to tackle a question from Brazil.
Yes. Right. So when you guys, got to reorganize myself here. When you guys did when you did the Brazil podcast, you had a letter from somebody who I thought was a brilliant letter.
Yeah, it was great.
Who asked, said, if you make antibodies to a protein, how does that work in a Western? Because in a Western, you denature the proteins. And so he was envisioning, he had this great analogy, Dickson, I don't know if you've listened to this, where he said a native protein he thinks of as an egg, and a denatured protein is scrambled eggs. I thought that was good.
But the
so
antibodies,
he's envisioning making an antibody to the native protein, and he says on a Western, as I understand it, is denatured. You got scrambled eggs. How's that same antibody going to recognize the protein on the Western?
And yet it does. All you have to do is explain how.
Right. So on the show, they explained it. Okay.
Yeah.
First of all, I want to say that we use the word all the time on this show, epitopes.
Yes.
Okay?
Yes.
Without ever having defined it.
That's
These are just things that antibodies attach to. And in the case of proteins, that's going to be probably sequences of amino acids.
A smaller portion of the protein.
Right. But in the case of proteins, they come in 2 forms. One's called linear epitopes, and that'll be a linear sequence of amino acids.
Yep.
Just sequence amino acids strung together. And the others are called conformational epitopes.
Correct.
And that's where you have in a folded protein, amino acids from 2 different parts of the protein that are contiguous. They're next to each other in the folded structure, and the antibody is recognizing amino acids from 2 different parts of the proteins that happen to be together on the 3-dimensional structure.
Right.
That's called a conformational epitope because it depends on the proper conformation of the protein. So if you had an antibody that recognized a conformational epitope, that wouldn't work in a Western.
Right.
All right?
Right.
And so you can try and engineer antibodies that will actually be specific for either conformational or linear epitopes. You can make a peptide to an antibody that will be presumably a linear epitope, or you can make an antibody you can inject the animal that you're making used to raise the antibodies with an intact protein, and presumably you might get something that's to a conformational epitope. You could also get out of that linear epitope.
Well, anti-idiotype antibodies are made against the pocket, which is a combination, right?
Exactly. So the point I really wanted to make is that this is all black magic.
Right.
Okay?
That's right.
And from a practical point of view, in the laboratory, you just sort of close your eyes and shoot up the animal. And then you get antibodies out the other end. And it's anybody's guess whether those are going to work in a Western or an immunofluorescence or an immunoprecipitation or an ELISA or an immuno-EM. You just don't know what you're going to get. It's a guessing game. And it's enormously frustrating. I've got antibodies that work great in Western and actually work in immunoelectron microscopy and don't work at all in immunofluorescence. Go figure that out. Okay.
Yep.
And so it's a very frustrating guessing game.
And then there's the sugar moieties on the outside of the proteins that could also elicit antibodies.
Sure, absolutely.
Yeah, it's black magic. That's what it is.
Black
It
magic.
truly is.
All right.
So I thought it was a great question, and we talked a lot about antibodies and this kind of stuff. I thought it was worth exploring in a little more
Good.
detail.
No, that was great. Thank you, Rich. It's a great question.
Yeah, great question. He had another question, too, that was great. I forget what it was.
All right, our next one is from Kelvin, who writes, I have just seen some of the ridiculous comments regarding the picture which was posted on your TWiV website. That must be TWiV,
that was
TWiV
the
150, exit
Right.
XMRV, or contaminated. Right. Yeah, contaminated. I've had CFS for over 16 years. I am a very firm believer in scientific method and have found it very frustrating at the lack of top-quality scientists who have taken an interest in well-defined patients. It has therefore been a great pleasure to see people like Harvey Alter, John Coffin, and others conducting research into XMRV. The data is the data. I hope resources and attention are now swiftly moved to other areas. I want to personally thank you for giving XMRV a good run. As a CFS patient in the UK, I'm used to seeing most apparently novel findings shot down rather quickly or simply not followed up. It has been heartening to see the XMRV issue tackled professionally and thoroughly. A true victory for science regardless of the unfortunate events now taking place, the slide issue.
Slide issue.
Sorry. Right. I'm sorry you have been on the receiving end of attention from some of the less balanced members of what might loosely be termed the CFS community. I think anyone with any objectivity and belief in scientific method would frankly lose the will to live after just 5 minutes of reading some of the material on MECFS forums. I don't know if you're aware of the background, but this forum was set up after a group of very rude and rabidly pro-Mikovits posters left the Phoenix Rising forums after continual warnings about their conduct. It's actually possible to spot their contributions on Deckoff Jones' blog and many other places on the internet. They're an extremely vocal minority who misrepresent the vast majority of CFS patients. I'm sorry if you've been subjected to this kind of nonsense. Apart from anything else, most CFS patients simply don't have the energy to sustain the relentless garbage which they produce.
Exactly.
My reading of some scientific literature over the last few years, my degree is non-science, so it's layman's opinion, suggests to me that immune dysfunction rather than a persistent infection results in the immunological manifestations of CFS. However, it would be great for TWiV if Professor Lipkin finds something in his pathogen project. Mm-hmm. It's just fantastic that someone of his caliber is doing the work, whatever the data. Once again, thanks for your patience and understanding. It's Kevin, not Kelvin. I'm sorry.
I was going to say, was his first name Lord?
Kevin.
Which, by the way, wasn't the actual name of that person either.
It was.
That's a beautiful letter.
No.
Thank you.
A really nice letter.
Yeah, it was a great letter.
And well written, too.
Yep.
I like that.
By the way, there's just a paper in PLOS ONE out of Norway showing that a monoclonal antibody called rituximab, which targets B cells, alleviates symptoms in a subset of CFS patients.
Really?
Yeah.
Interesting. Yeah, I think this idea that it's some sort of immune dysfunction makes all kinds of sense. And that would be consistent with that.
Rich, why don't you take the next one?
Okay. Janet writes, Hi, TWiV gang. I just wanted to let you guys know that I love listening to your podcast. I only recently discovered it, so I'm still trying to frantically catch up with all the episodes. I remember you guys asking for a transcriber back in episode 30-something, but judging from the transcripts section of the website, I guess there weren't that many takers. I don't know now if you're still taking transcripts, but I thought I'd try my hand at it, so I've attached the transcript of the latest episode.
Oh, wow.
I couldn't figure out the formatting for the header, so I've also sent the doc file to play around with. I guess I can see why there aren't so many people jumping to volunteer for this, as it took a lot longer than I thought it would. Nevertheless, if it's all right with you guys, I will try to transcribe a few more episodes, especially the Virology 101 episodes, because I feel those would be the most beneficial to both the audience and myself. And we should do more of those.
Yeah, we should.
I'm starting my PhD in 2 weeks at the University of Heidelberg in Germany.
Marvelous.
It is a charming town, and I will be working on foamy virus vectors in the same building as Professor Harold Zuhausen. So I'm very excited. It would be great to see an episode on foamy viruses one day. You mentioned them once briefly in the reverse transcription episode, and I find their apathogenicity and therapeutic potential fascinating. I'm coming from a biochemistry background, so I hope I'm not getting in over my head, and any advice you could offer on the PhD life, virology, or science in general would be fantastic and greatly appreciated. Keep up all the good work. Grüße aus Deutschland, Janet.
Well, my advice is don't get into any sword fights.
I trust that, I presume that she is German.
Right.
I mean, that's an assumption. In which case, it's phenomenal to me that she's transcribing episodes into English.
Yes.
But clearly, she's virtually she's quite fluent in English. At any rate.
So she sent us a transcript of Symbiotic Safecrackers.
All right, great.
And it's up. I put it. Thanks so much, Janet.
Neat.
And I will repeat the offer I put on the website. Anyone wants to do a transcript, I'll send you a TWiV mug.
Hey.
I know it's not a lot, but it's our token of gratitude.
Whose mug is it? Is it Alan or you or Rich or myself?
Good.
That would be good, Dickson.
Good.
I could put your picture on the side. Of the mug.
We should put our pictures on the mug, though. We should really do that somehow and encapsulate it.
Nobody wants them.
So I had a conversation with somebody about foamy viruses recently, and for the life of me, I can't remember who it was, but they are fascinating and a good. It'd be nice to have a lecture on those. And yes, Heidelberg is absolutely a delightful place.
Richard, why are they called foamy viruses? Just in passing, because of my naivete here.
I forgot.
It's the way they make cells look. They make cells look foamy when they're infected.
They they make them
it's
like
their
that.
cytopathic effect.
Yeah.
Okay.
Very interesting viruses. We should do an episode. We get someone who's an expert on foamies, and they're very
Looks
cool.
like we have a burgeoning expert right here.
And Harold Zerhausen, by the way, is a virologist of significant repute. In my mind, famous, among other things, for making the association between papillomaviruses and cervical carcinoma.
Oh, wow. Big one.
Our next one is from Zachary, who writes, I'm a nursing student at the University of Washington and love listening to TWiV on my commute. In my pathophysiology class, my teacher recently told us that the problem with polio is that immunity is not lasting as long as we previously thought. She stated that we are now seeing increasing cases of polio amongst the elderly due to the decrease in memory T and B cells. I've listened to several of the podcasts on polio and do not remember hearing this information. Can you please enlighten me on this topic? Thank you, and keep up the amazing podcasts. Well, first of all, Zach, there isn't any polio in the U.S., so I don't know what your teacher was referring to. And the polio that is in the rest of the world, it depends where you're looking, but in many places it's in kids. There was an outbreak recently in slightly older individuals, but these are mainly non-immunized people. So there's nothing to do with immunization lasting a long time or not. As far as we know, immunity to polio is lifelong against the serotype that you're infected with, and that goes for the vaccine as well. So I know of no information that indicates what you're saying or what your teacher said was correct, that it's increasing among the elderly due to the decrease in T cells and B cells. It's simply not the case. I mean, we just can't immunize enough people to stop the spread. That's the problem. It's not because there's waning immunity. What do you think, Dickson? Rich?
I don't know anything about that one.
Yeah, you know more than I do. The only other thing I can suggest is I would be interested for this individual to approach his teacher and say, okay, so where'd you get that information?
Yes, that's a good point.
Now, wasn't there an outbreak in Minnesota, Vince, among some religious group?
So a number of years ago, there were 3 or 4 cases of polio in an Amish community. They're actually virus isolations from children, and they were vaccine-derived polio viruses. And more recently, an older woman died of polio that she had replicating in her for 12 years. She was immunodeficient, and she acquired the vaccine virus from her child when her child got polio 12 years earlier. But anything to do with this. But Rich has a great point. Zachary, ask your teacher for the data.
Yeah,
Where is it?
That's right.
Yeah, that's a good point. All right, we will do one more here. Rich, take that last one.
Okay. Catherine writes, Dear TWiVers, I am an ESL teacher in South Korea. I discovered your podcast through iTunes about a month ago. Though I now teach, I have a background in science, college, and a master's in psychology. I enjoy your podcast greatly as it appeals to my math, science dominant side. I enjoyed your recent episodes on XMRV and how well it demonstrates how the scientific process and peer review work. I was additionally interested because I was recently diagnosed with fibromyalgia, which seems to be related to CFS, at least at the symptom level. Do you know of any work being done on possible viral causes of fibromyalgia? I also have a quick story to share. At an ESL training yesterday, we played essentially Taboo as an example of an academic activity. We can't call them games here or the parents go nuts. One of the 10 words my team wrote down was phage. The team that got our paper passed on it. The leader asked if anyone knew and then asked whose word it was. I meekly raised my hand, admitting that it was mine. And she says, well, what the heck is it? We all want to know. After saying it was a virus that infected bacteria, someone replied, you weren't an English major, were you? That's where I admitted, no, I wasn't. Thanks for your show and keep up the good work.
Fantastic.
So we're useful for all kinds of things.
You bet.
Right?
That's great. I don't know of any...
She was unfazed by the question.
Jeez.
I don't know of any virus associated with fibromyalgia. People are working on it. They looked at XMRV. There were a few publications, but found no connection. And, of course, now we know that XMRV itself couldn't be causing it because it's a laboratory-generated virus. There are if you look in the literature, there are other papers attempting to make a connection, but nothing convincing as far as I can tell. But we do need to have some studies such as the one Lipkin is doing on CFS, I would think. Mm-hmm. I don't know if those are underway or not. Let's do some picks of the week. Rich, what do you have for us?
I've been saving this one up for a while. I want to point out Google's self-driving car. This has been going on for a while. Several years at least, Google has been developing a car that will drive on regular old roads without a driver. And it's fascinating. And I've got a couple of links in here to two different fairly recent articles on how the thing works. So it's got a whole bunch of sensors on it, a big laser thing on the top, and a bunch of radar bits on the sides. And what they do is they take it out and drive it on the route that they want to take it on. And all of these sensors basically look around and create a map of the route that they're driving. And that map, there's got all kinds of fancy algorithms and stuff so that the map can differentiate between fixed objects and stuff that's going to move. It can see stoplights. It knows what color they are, all of this kind of stuff. And then they can take the car, and of course they actually have a driver in it sitting behind the wheel, but he keeps his hands and feet off, and they drive this course, and it'll drive from L.A. to San Francisco.
Darn.
Just, on the highway, on the side streets, you name it. And it responds to pedestrians, it responds to traffic lights, it responds to other traffic, all this kind of stuff. And it's actually not had... The only accident, I think, so far is somebody rear-ended one of these.
Of course.
So they haven't hit other cars or pedestrians or anything?
No.
Trees?
No.
It stays right on the road where it's supposed to?
Yep.
Wow.
It's amazing.
So you think someday we're going to just kind of sit in the back of a car and it's going to drive us wherever we want?
Yeah.
Eek.
And the idea is that might be safer.
It would be.
Okay? The car's not going to get drunk, among other things.
That's true.
What if it gets a virus?
Ooh.
A computer virus.
Yeah.
The kind that kill you.
So it would require, at least with the current technology, it would require that there is a a map of this type of all of the roads that this car is going to drive on.
I
All
see.
right, and that seems like a massive task. But consider, Google Street View. Heck, they're already driving all over the place, making maps, and so it's not out of the question.
So does your son know about this? I guess he does.
I'm sure he does. I haven't questioned him about it.
I wonder how enthusiastic or positive they are about this becoming a reality. I mean, are other companies doing this too?
Not that I'm aware of. It's hard to imagine that somebody else isn't, but it's, when you work for Google, they tell you that they want you to spend 20% of your time doing something that's not actually your job.
That's right.
Okay?
Yeah,
and that's where all this crazy stuff comes from. One of these articles in it has a little video that is a presentation by a couple of the Google guys on this project, and you can see the enthusiasm in them for this. You can see I mean, this is so geeky.
Cool.
It's crazy.
Hey,
That's cool.
planes fly themselves, right?
Yeah, absolutely. Absolutely.
The thing that's going to happen here is someday we're all going to have these computer cars. They're going to become sentient, and then they're going to drive us all off the cliff. It's going to be the end of humans.
One of my future picks is going to be Battlestar Galactica. That's
Right.
where that's going.
Okay, that's pretty cool.
Funny.
Dixon, do you have a pick?
I do. I have several picks.
Well,
They're
you
related.
can't have several.
Yes, I can. I haven't been here for a while.
Oh, okay.
No, they're related to each other, and I've been absolutely glued to my television set as a result. One of them is called Wild Russia. It's a series, and the other is called Wild China, which just started, and they are amazing. They're like Planet Earth, but only on specific areas now. And the Wild Russia was, I mean, when you realize how big Siberia is, right? It's 3/4 of Russia. And if you look at what's encompassed in that, I think they said that there are 18 time zones in Russia. That's an amazing number of time zones, yes? I mean, it's an enormous amount of land. And when you look at these maps, the only maps we ever see are the ones that have vegetation or, you don't see the countries because if you look at Siberia, it's all included into the Russian country. You don't know what's there. And this program takes you from time zone to time zone and shows you the natural life that still exists in these places.
Cool.
Now, the one I saw last night, because I just got back from Shanghai, so I'm a little bit jet lagged, so I was up at 3 this morning watching the first in the series of Wild China. And, of course, they went to Tibet as well because they didn't consider China and Tibet as the same country, although there is some controversy here because of the Chinese will to take it over, I think. But Tibet was shown to be the cause for the monsoons. Now, this is a dry, hot place in the summer and a very dry, cold place in the winter. How can that kind of environment influence weather patterns on the other side of the Himalayan mountains? How does that work? All right? And they showed very clearly how this works because when this thing heats up in the summer, it's like a big hot plate. And you can imagine all the hot air rising up into the atmosphere and creating a circulation. And when hot air rises, something has to come in to fill it in. And what is it? It's the air coming in over the Indian Ocean. And it picks up all this moisture. And so it's being driven up the slopes of the Himalayas. It, of course, never goes to the other side because it's dry as hell over there. It all gets dumped on the Indian side of the Himalayan mountains. But to explain Tibet from the perspective of world weather patterns, that was, for me, that was the biggest take-home message I've ever heard. So
Cool.
it's going to have lots of those situations in there. So I'm just fascinated by it. The photography is excellent. The narration is spot on. It's a great series, and I highly recommend it.
Excellent.
Thank you, Dickson.
You're welcome.
Let me get over that jet lag.
It has nothing to do with viruses, by the way.
It's all right. Science. It's
Yeah,
science pick of the week.
Well, I hope it's good science. I hope the narration is reflective of the amount of studies that went into the statements.
My pick is a book by George Rohrman, who is a virologist out at Oregon State University. I met him down in Brazil. He came up to me and he said, I wanted to say hello. I really enjoyed your keynote and TWiV. And he gave me a book that he wrote. It's called Baculovirus Molecular Biology. And he gave me a copy. But he told me it's available for free at the NCBI bookshelf.
Really?
I don't know if our listeners know about this, but National Center for...
Biological Information.
Are you sure? All right. NCBI has a bookshelf where they have many free books. Many of them are old, but this one is brand new. He just wrote it this year, and it's a wonderful summary about baculoviruses. So he had printed out some copies. He wanted to get rid of them because he didn't want to carry them back with him. He gave me one, but this is free to everyone. So check it out. You know, we've talked about baculoviruses here.
Yeah.
And I just want to point out one passage. I started reading it, and just in the first page or so, in one study, it was found that cabbage purchased from 5 different supermarkets in the Washington, D.C. area were all contaminated with baculoviruses to such an extent that each serving about 100 square centimeters of leaf material would contain up to 10 to the 8th polyhedra of a baculovirus pathogenic for the cabbage looper.
The cabbage looper.
Which is a bug that eats cabbage, right?
So this has been spread onto the cabbage by
the bug. And we eat it.
No,
the cabbage is raw, right?
Have the farmers used this as a control measure?
No, they have. It just kills the...
All right.
It's not used as a control as far as I know.
All over the... Well, I think that baculoviruses have been at least considered, if not actually used. I think they've actually been used as biological control, but not this particular one. Doesn't matter, man. You're eating baculoviruses all the time.
You're eating, and that got me thinking about the viruses in your food, and that's why I looked up the viruses, the plant viruses in your feces. So,
very cool.
So it's a cool book, and it's free. You can go download the PDF, and it's written this year. It's up to date. George is a good guy. Thank you, George. And there's my pick of the week.
Fantastic.
NCBI is National Center for Biotechnology.
Okay.
There you go.
Not bad.
Thank you.
All right.
Close enough.
We have a listener pick of the week from Jim. Vince et al. Lee Cronin gave a TED Talk on making matter come alive in July 2011. He describes the steps in the development of life from matter, including how cells are made. I think this has interest because of the ongoing debate on whether viruses are alive. It raises the question of when life starts as molecules aggregate. 73, Jim.
Did you have a look at this?
Yeah, it's pretty cool.
Yeah, it's pretty good. He's studying the notion that he can have inorganic molecules that basically self-replicate in compartments built of other inorganic material.
You know what 73 means, Dickson?
I haven't got a clue.
It's a ham radio thing.
I don't know that.
You don't know
Yeah,
what
it's
it
kind
means?
of like, do you?
No,
I mean, Alan told us at one point, right? Do you remember, Vincent? It's kind of like over and out or something.
Means my love to you.
73.
You know, it's like a greeting.
All right.
A friendly greeting. Thank you, Jim.
I know what 86 means.
What does that mean?
It means get out. You got to 86 it.
Really?
Yeah.
In what language?
I have a feeling it's CB.
I thought it was we're out of that in restaurant language.
There are many numbers that mean things in different languages.
There are.
But we can't talk about them here on TWiV.
No.
Unfortunately.
No.
But when we're done, I'll tell you one that's very good. That'll do it for TWiV. Don't forget we have a fan book. No, not a fan book. A Facebook fan page. Facebook.com/This Week in Virology. Go fan us or friend us or whatever it's called. And we post things there now and then, and you can too. You can find us on iTunes at the Zune Marketplace. At TWiV.tv, we also have a phone app for your iPhone or Android device at microbeworld.org/app. And we do like if you write reviews at iTunes. If you're brand new to us, please go over and just write a sentence. It helps keep us on the front page of the iTunes medicine directory so more people discover us. And do send your questions and comments to TWiV at twiv.tv. Dickson Despommier is at verticalfarm.com, Racaniello.org, and what's the other one?
Medicalecology.org.
Thank you for joining us today, Dickson.
It's my extreme pleasure.
Good to see you.
Yep.
Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
Sure. Great time.
Always a good time.
Always a good time.
My name is Vincent Racaniello. and you can find me at virology.ws. You have been listening to This Week in Virology. Thanks for joining us. We will be back next week. Another TWiV. It's viral.